vitro BE approach?
So our goal is to ensure that characterization-based approaches can be made available for any topical dermatological product. So depending on the specific corticosteroid product, yes, a characterization approach is feasible.
The PSGs for the topical corticosteroids don't reflect that specifically, but yes, the characterization-based approach. Topical corticosteroids are not somehow special in that way compared to others. So the short answer would be yes.
All right, microphone number 2, nice and close for me, please.
Hi, good morning again, and wonderful presentations. I was very interested in the Q3 similarity. In one of your slides, it shows in order to demonstrate Q3 similarity, you have to show that your test product range falls within the range of the reference product. So my question to you is that how do you demonstrate, how do you determine the range of reference product? How many batches do you test, or do you need to test a different reference product of different ages?
The expectation that is laid out within our PSGs is characterizing at least 3 batches of the reference product, if 3 batches are available, and determining the range or the variability that you observed by using that data. Now, what Dr. Ramezanli referred to in terms of aged versus new batches, it could happen, say, for example, for a specific Q3 criteria, such as particle size, that the particle size in the product is growing over time. In those situations, if you compare the newly manufactured generic product to, say, for example, aged sample for your reference product, and they compare well, they fall within the range, we don't know for sure if for the generic product also the particles can grow over time, and in those cases those specifications will fall out of that range, and therefore we recommend in those situations, if you do observe a trend within the reference product or expect a trend for your generic product, that you compare batches that are across the entire shelf life duration.
Thank you.
Sure.
Microphone number 1, please.
If you have leachable samples that are available at the time that you're going to do your extractable studies, can the 2 studies be combined to have efficiencies?
Yeah, thank you for the question. The question is, can you combine these extractable studies with the leachable studies? I would say, the extractables kind of has a different purpose than the leachable study, and you're kind of using that as like getting a broad picture of what could potentially be present, and making sure that your methods can detect it. And again, there you're using a wide range of solvents, right? It's not just one solvent, because you kind of want to get this more comprehensive picture for your product understanding. So then the leachable is more focused, where you're looking at the finished product rather than individual components. And then based upon the extractable work that you had done ahead of time, you kind of know what you're looking for, and then you can, more adequately design the leachable study according to that. So I would say in general, we would expect them to be 2 separate studies. Yeah.
Thank you.
Yeah, you're welcome.
Online question, please.
When validating an IVRT study, what altered formulations should be used to show sensitivity, specificity, and selectivity?
So during IVRT validation, the general expectation is that during the validation steps, you are using altered formulations that are at 50% of the strength compared to the reference strength, the product, actual strength of the test product, and then at 150% of the strength. And specifically focusing on the higher side, we recommend that you don't go beyond the 150% strength, because we have seen some examples where a product has a 200% strength, or say, for example, for a cyclovir topical cream, 10% cream is being used to validate the study. And our assessment is if the IVRT method is unable to discriminate a 7.5% strength from a 5% strength, there is no way to establish that the method is sensitive enough to evaluate or discriminatory enough to evaluate the compare the test and the reference product. Therefore, at the higher end, we definitely don't recommend going beyond 150%, and you can use a lower strength sample if you have limitations related to solubility and some other considerations.
All right, microphone number 2, please.
So thinking of efficient science-based studies, do you foresee using just VC studies to approval of 505(b)(2) topical steroid product?
Your question relates to how you would approve a 505(b)(2) product. But I'm not sure that I have enough information based upon what you've said. That I think you said it's a steroid and you might use a VC study, but I think I'm not sure that I can answer that question based upon the information you have.
So I was thinking, can I use the VC studies for efficacy to file a 505(b)(2)?
I'm not sure. I think that so the 505(b). So this focuses more specifically on situations where the talks we've been focused on really are on generics where the products are pharmaceutically equivalent. So in a 505(b)(2) situation, your product may not be pharmaceutically equivalent, and then depending upon what the differences are, I'm not sure whether a vasoconstrictor study would be appropriate there. That's why I'm not sure, how different or what the basis is or what the reason is, what's different about the product in your 505(b)(2) situation. And that's why I think I might.
The recommendation for a 505(b)(2) is that he request a pre-IND meeting with the correct OND division for which the product resides, which could be the derm division.
Details about your specific product, I think, would really help to identify what types of evidence would be appropriate.
Okay, and we'll stay right here at microphone number 2, nice and close, please.
This relates to the change in the design of a TDS system, what you mentioned. In case what you showed as an example, a spherical versus a rectangular, suppose, versus a spherical RLD, a rectangular test product is developed. And if that test product, suppose it fails in adhesion in one orientation, say horizontal, but passes in vertical, would that be acceptable? The second point is, in this case, since it is considered as a drug-device combination, would you need a human factor study as well?
Okay, so I can speak to the quality aspect. So if you're changing the shape of the system, we would expect that the size would remain the same, like the surface area still should remain the same.
Yes.
And then you would need to update your batch records accordingly to the different size. But from the quality aspect, that's relatively simple. I think on the bioequivalence and clinical aspect, there's more to it.
So to address your first question, if the product fails adhesion in one orientation, it may not be acceptable because we cannot expect a patient to read the product label and apply the product in a specific orientation, specifically when they are used to the RLD product and it's spherical and you can basically apply it in any orientation.
If you clearly mention in a label the way it has to be applied vertically and not horizontal.
In those situations, I think that the second aspect that you brought up may be a useful aspect, looking at human factors and understanding if the directions can be followed. But I would still say that that's a very this is a failure mode that we would take very seriously if the product is falling off when you are applying it in one orientation compared to the other, and the expectation that patients would read and apply and follow the directions exactly is relatively high standard, specifically when there are design differences between the test and the reference products.
Understood.
There are
Thank you.
comparative analyses that can demonstrate that certain differences may be acceptable. But I think an underlying consideration there is that patients or caregivers should not now need to be retrained on how to use the product because of a difference in the or a potential different failure mode for BE or for TE for the generic product.
Thank you.
Online question, please.
Why do some PSGs for topical products include both IVPT and PK studies? IVPT, simulated PK study in vitro. Why do we need IVPT if we are doing a PK study?
I understand the question correctly. It's asked why we recommend IVPT study in some PSG and PK study in some others in addition to IVPT. So as we mentioned in our presentations, the studies that are recommended depend on the complexity of the product. So when we have a multi-phase system of semi-solid and depending on other aspects of the complexities, we may require to get information about cutaneous pharmacokinetic if applicable. And also it depends on site of mechanism of action. Sometimes there are evidence supporting that there is systemic site of action in addition to local action, site of action. So we would recommend PK studies as well. But PK study give us different information, give us more information about systemic pharmacokinetic, not cutaneous pharmacokinetic. So in some cases, in addition to IVPT, we may need to PKB study, and PKB study cannot replace IVPT.
All right, microphone number one.
Hi. I have a question on the leachables and extractables requirement. I want to understand if the same standard of review is applied for the transdermal systems and the oral systems in terms of the leachables and extractables. When it's a fact that skin is the biggest barrier for absorption of the molecule itself, the active molecule itself. What is the stand on?
Sure, yeah. Thank you for your question. So if I understand the question correctly, so you might disagree with the transdermal route being treated differently for extractable leachables than the oral route. There could be barriers to absorption based upon the size of the leachable, for example. So what I can tell you is this is our current approach based upon the understanding across multiple groups within the center. And so this is how we are currently approaching transdermal systems. And as we continue to gain more information, then I think we will try to update it according to what the science would demonstrate.
Thank you. Thanks. Okay, microphone number two, please.
Thank you for the presentations. And this question is related to the bio-guidance that is referenced in most of the PSGs. The bio-guidance typically calls for Cmax, AUCT, and AUC infinity. But for transdermal products, does it make sense to ask for AUC infinity because you're taking off the patch? If that is the last sampling point, once you take off the patch, there's no absorption, and whatever is there in the skin or in the systemic circulation is what is comparing between brand and test product.
K metrics that are recommended, we believe is the most appropriate PK metrics to be evaluated. And even for some of the PSGs, when we find out that the partial AUC is relevant, we even ask for additional PK metrics such as partial AUC, if I understand your question correctly.
My understanding is AUC infinity. Is it a requirement for transdermal products to have AUC infinity equivalency? Because the guidance is mostly broadly, you're referencing a guidance that is meant for most of the dosage forms, and especially solid dosage forms.
I think the recommendations within the guidance are applicable broadly to any systemically acting products. And since transdermal systems are considered systemically acting products, in addition to looking at the absorption phase, we are also interested in understanding the elimination phase. and if those matches for the test and reference product. So we do recommend looking at all the three parameters, so Cmax, AUC 0 to T, and AUC 0 to infinity for systemically acting products.
Thank you.
All right, our next online question, please.
What information should we provide to justify upper and lower limits for proposed specifications on physicochemical properties like apparent viscosity?
So I think for viscosity, or more importantly, when we talk about semi-solid products, we are more looking at rheological assessment. And the general expectation is that the limits will be set based on comparing the whole rheological profile for the reference product to the test. The ranges can be set based on the variability that is observed for across the 3 batches of the reference product.
All right, and we do have the panelists for about 10 more minutes, so if you have any questions in the room, come on up to the microphone, but let's go ahead and take another online question, please.
And the BE approach for a dual active drug product, as in the example, would a single IVRT and IVPT study that characterizes both drug products be required, or would it be acceptable to have separate IVRT, IVPT studies for each individual drug substance? 2 IVRT methods and or 2 IVPT studies.
So if I understand correctly here, you're talking about 2 drug molecules in the formulation. So if that's the case, then yeah, we would expect 2 separate IVRT methods. I mean, for 1 IVRT method for each drug substance, and characterize both release profile, and the same with the IVPT, characterize the permeation profile for both of the drug substances. And if your method can distinguish, if your method can detect both of them, then you can use 1 IVPT method, correct? Or the same with the IVRT.
So, the point is that the bioequivalence needs to be demonstrated for both active ingredients, both in terms of the release rate as well as the permeation. And if a single IVRT method can evaluate and be validated to evaluate the release rate of both compounds, and likewise in a single IVPT method that your analytical method can do the same, then that's not a problem if it's possible.
Okay, microphone number 1, please.
Okay, this is regarding leachables and extractables for the transdermal. Can we apply the risk-based approach when we have the initial extractable data and 24 months leachables data on 2 batches, and go with the risk-based approach and demonstrate when we don't have the multiple points during the leachables data, and as a post-approval commitment kind of thing?
Yeah, so thanks for the question. You know, in general, I would just say, you know, kind of what I demonstrated on our expectations, those are like what we recommend if you're starting today, right? The ideal situation. You know, if you're able to provide sufficient information to qualify the safety, such as you've outlined, you know, you have the extractable study, you have data at release and near shelf life. So that may be sufficient, but that would, you know, of course, be determined during the assessment. And I guess, did that address all of your question? Yeah. Okay. Yeah, thank you.
Okay, another online question, please.
I would like to get a clarification regarding comparative dissolution testing for transdermal TDS. Is comparative dissolution in different pH still required to obtain biowaiver based on the dose proportionality?
No, we don't. We no longer recommend conducting the dissolution studies at multiple pHs for the requesting the waiver aspect of release studies.
Got another one for me, Ray?
How do we report water quantity in batch formula and in 3,2P1 for the water overage used to compensate the water loss during the manufacturing process?
Yes. If you need to use overage of any ingredient, we in the formulation table that you submit, we want you to report the nominal amount, and then maybe in the footnote you can clarify that you're using overage of any of those ingredients.
Okay, we've got about 5 more minutes. If there's any more questions in the room, another online one, please.
The PSG says to do a clinical endpoint study, but a sponsor is proposing Q1, Q2, Q3 approach combined with IVRT and IVPT. What data needs to be submitted while requesting a pre-ANDA meeting?
I think if I understood the question correctly, you're proposing an in vitro-based approach, and the question is what kind of data may be needed. So I think, like Dr. Ramezanli mentioned during her presentation, providing data about the formulation, detailed information related to the each of the excipients, as well as your Q2 aspect of the formulation is helpful. Then providing Q3 characteristics, as much data as you have related to, say, for example, rheology, pH, the manufacturing information for your proposed test product. For IVRT studies, providing at least a preliminary method and comparative release from the test and reference product. And feasibility studies for IVPT would be ideal. We have accepted meeting packages which have not contained all of these data in the past, but that makes the determination and a feasibility assessment of the proposed approach really difficult. So if available, this is the level of data we would recommend for a meeting package.
Okay, and we've got time for one more online question, please.
During an extractable leachable study, if transdermal patch is exposed to major harsh solvents and cannot find any leachables, do we still need to test leachables during stability studies of exhibit batches?
Thanks for the question. You know, when you're doing the extractables, you should be getting things out. I would say, you know, there's components in these different polymeric materials, so you should expect to see something coming out. So again, you're wanting to target, you know, different solvents, and then make sure your extraction conditions are acceptable, so that you are kind of seeing. And then you've also talked with your vendor and got an idea of what's in there. So, you know, we would still expect, you know, let's say you did the harshest extraction possible, you're not seeing anything, we would still expect to see a leachable study then also provided.
Okay, and that is all the time we have for questions. Please help me thank our panel and our presenters for coming to talk to us today.